Hello students, welcome to the MOOC course on bioengineering. I hope you are able to see me and hear me now. So I'm right now in my office at IIT Bombay, and I thought to make it an interactive start in informal manner with you. Let me say again hello and welcome to all the people who are joining in for the live session. I'm Sanjeeva Srivastava, Associate Professor in IIT Bombay in Department of Biosciences and Bioengineering. I'm currently in my office conducting this live, informal, interactive session with all of you. So, of course, you can post some queries on the forum if you want to ask some questions to me. Uh, First one, I just thought to tell you what this course is about. Of course, you have started viewing some videos during the first week, so you got some idea that what kind of contents we have for this course. So just think about many things which we want to learn. They can be of common interest to humanity, right? So irrespective of our discipline and our specialization, whether we are coming from biology background or engineering background, lot of technologies and lot of solutions which are required, eventually they can help the humanity. So this course is essentially trying to bridge the gap, fill that void which we have between different disciplines of biology and but medicine and engineering disciplines. Ideally, if you think about, we have to make some choice at some point of career. We want to go to the engineering route or we want to go to medical route, right? But at the same time, when it comes to the life problems, then we have to think about overcoming those boundaries, because the bigger problems are not going to distinguish things based on biology or engineering. So therefore, we need to understand the fundamental problems. We also need to apply various engineering technologies and solutions to resolve those problems. So many times for the same given problem, we have to use interdisciplinary approaches, right? So that's what the intention for this course was: how to convey you some basic concepts of biology, in which way technologies can provide more solutions to it. And if let me kind of give you a quick look into this video contents for this week. So the first two lectures, I have tried to give you various examples and trying to emphasize why to take this course on bioengineering. Many problems I have tried to give you illustrations, whether it's environmental related, healthcare related issues, and those problems are so pertinent for all of us. So I try to give you some idea that in which way the technologies have made lot of contribution to the medicine and biology field. After those two lectures, then I thought to talk starting from the basic fundamental units, which is cell. Uh, and then the various ingredient, various components of the cell, different organelle, what role they have, in which way we have the broad taxonomy based on the prokaryotes and eukaryotes. But again, intention was not to give you too much in depth of biology, but rather give you the feel that in which way biology works. So some very basic glimpse of the basic concept for the cell and various cell properties. But again, the motivation and the intention for this course is not to make you a biologist, not to make the course very heavy from the biology contents, but rather give you some basic concepts of biology and then try to give you feel how technologies are making an impact for various type of solutions. So the last lecture of this particular week, I had invited one of my clinician collaborator, Dr. Ali Asgar Moyadi. He is the neurosurgeon at Tata Memorial Hospital. And Dr. Ali gave you an idea in which way the neurosurgeons, during the surgery, they look for a lot of solutions, right? Again, various type of indigenous devices, various type of, precise ways of looking at things for the surgery can be so helpful for the clinicians. So that also gives you some way of connecting with the healthcare and technology aspects. So all this, the first week was very introductory, where the intention was to give you why you need to take this course, what kind of things one could learn from the cell, and why even clinicians they need interface with biologists and technologists together. So that is kind of the week one. But as we go along and we progress in the next week, so in the second week, the major discussions will happen around understanding about the nucleic acid, knowing the DNA, and how various tools and technologies are being used to study DNA. Various type of latest revolutions which are happening around us, all of us in the clinic, in everywhere, is based on the DNA based testing. Various diagnostic things are happening based on polymerase chain reaction, based on next generation sequencing, and many things which are happening. They're all technology driven, technology oriented things. So again, how to study these DNA and these basic molecules of life, and how to apply these technologies is something which we are going to talk in much more detail in the second week. What I think important to convey here, that while you are taking this as an online course, but I still want to give you feel that we are pretty much available to you in person like a classroom teaching. Although there is no substitute of classroom teaching, but still I'll make it very interactive. Every week I will be available at least once in a week to interact with you in this kind of live sessions. Of course, courses are being taken by thousands of students, so I cannot reach out to each one of you personally. But at least in this kind of live stream session, I'm able to reach out to hundreds of thousands of students who are taking this course. So then you can put your queries, and what we can do, we can also conduct a Skype session, which we are planning to conduct every Saturday. So not this Saturday, but from the following week, every Saturday we will have a live session where we can take your questions and queries, and those questions can be addressed. So if you can send your queries and your Skype ID by Friday, then we will be able to come back to you and have a session with you on Saturday morning. So this is how we are going to progress in the subsequent weeks. Again, my intention was to make it more informal. I don't want to give you another lecture after watching so many video lectures already. So let me give you the glimpse of my lab, where I work, what I do, and kind of some idea that how we are working on different technologies, especially for the clinical research. So I will move around the laptop and I will shift to our one of the laboratories. All right, so I have now come to my one of the labs where it is basically mass spectrometry based lab. Uh, Behind me, you can see various gadgets, various instruments, uh, which are heavily used for various type of proteomics research. Uh, so again, the area of research what I work on is looking at all the protein molecules in a clinical sample type. How various type of proteins they govern diseases, they govern various types of physiological action. Those things we want to learn, understand by studying the protein molecules in the field of proteomics. So these kind of instrumentation and technology, they are able to resolve the complex mixture of the proteins, and then we try to analyze those details and try to find out whether we can obtain the quantitative information that how the proteins are changing in response to a given disease, in response to a given stress condition, in response to a given drug therapy. So those kind of changes which are happening very fast inside the human physiology, how to make use of those information at the protein level, we can try to detect those, analyze those at the these kind of technology level, and this is what we try to study in proteomics. So again, while I'm talking, I'm not going to give you much more detail about what these instruments are about and what they do exactly, because these are very high-end technologies which people use mass spectrometers for various type of research. We will be coming slowly to you. We will be teaching many of these technologies as we go along in the subsequent lectures. But currently, as we are talking in the basic part, in these weeks we are talking to you about the cell biology concepts about the cell. Then we are talking to you about DNA molecule. We'll build your concepts and fundamentals about DNA technology. And then slowly we will take it from there and talk about the complex molecules like protein and various protein technologies. So of course you will have much more interaction on these instruments and these sophisticated technologies to know that how we can use them for various type of clinical research. So in addition to teaching various courses at IIT Bombay, the major research in my group revolves around clinical proteomics theme. One thing which we are very keen on looking at the brain tumors, how different type of brain tumors get aggressive, what kind of protein biomarkers we can identify to distinguish them based on their grading, looking at what kind of patient survival we can predict, what kind of therapeutic responses these patients may have. So all those information we try to obtain, looking at the proteome level, looking at all the possible protein from the complex tumor tissue sample, a cerebrospinal fluid, the serum sample, and many of these kind of biological sample which gives us the insight for doing these kind of proteomic research. Again, many of these things becomes very much technology driven. It becomes very much data driven. Big data is generated. We face sort of issues about where to store data, how to analyze the data, which kind of databases to be used, all kind of things which people are facing in different disciplines. But again, in healthcare and biology, we also face these kind of major issues about big data handling and big data analysis. Again, things become very exciting. Just imagine that once you have learned some of the basic concepts here in this course, you can access lot of data, lot of publicly available data from many repositories. Whether we talk the things here, from my laboratory or from many parts of the world, people are making lot of data available for various research for sharing. Now, if you know the tools how you can analyze the data, you can start downloading and accessing those information and start putting your hypothesis, and then you can take these things forward and come up with some very innovative solutions. So this can be just a very exciting start for us, and then we can think about how best we can make it more interactive, because our intention is to not give you just video lectures, but also give you feel that we are with you to take your curiosity, to take your quest for knowledge, to take your interest for venturing into biology field to the next level. And we are pretty much available for giving you, helping you out all the possible ways, and giving you demonstrations from the lab sessions as well as personal interaction with me. So, as I mentioned, every week I will try to conduct one live session where all of you can directly interact with me. Even now, if you have certain queries for me, if you want to know something, please do write the questions on the forum. If I can see that quickly, I will be able to come back and answer those queries to you. So this can be a good portal for us to interact. Although I realize it is one way of interaction. I can't see you, I can't hear you. All I can say that a lot of people are watching this video right now and interacting with me, but I don't have your way of interaction. So you can feel free to send me your queries, and eventually, as I said, every shuttle day, we are going to have the Skype session, technical part where you can ask us your questions. If you're not clear about something from the assignments, from the the lecture modules, if something is not clear, you can post those queries to us, and we will revert back to you and have a session with you on Skype. So I really wish that this course is going to serve the objective for which you are enrolled. This course is going to be interactive and interesting for you. Every week we will have certain sessions with the clinicians. You will have some live video sessions. Not in this manner the way I'm interacting, but in the lectures you will have certain modules with all the clinicians every week, and those will be very much eye-opening for you because they will give you the practical difficulty which doctors face in the clinic, and in which way technologies can try to solve the problems. So let's make it more interactive course, and I hope it will become very enjoyable for you. So I'm stopping this short interactive session now. I'll be happy to take any query if you have. Please put those in the on the portal here. And again, I look forward to interact with you subsequently in the following weeks. Okay, thank you very much and welcome again and join this course. I'll be very happy to interact with you over the period. Thank you.